go over the topic mass transport in animals, and this is part of the unit 3.3.4 of the new biology A-level specification for AQA. So we'll start off with haemoglobin. This is the stuff that actually carries oxygen around the body, and it's present in the red blood cells. Okay, so haemoglobin is a protein and it has a quaternary structure. So for some recap, a quaternary structure means that there's more than one polypeptide chain. So in haemoglobin there are 4 different polypeptide chains with an iron in the center. So each of these polypeptide chains can carry 1 oxygen molecule, and so the whole haemoglobin molecule is able to carry 4 molecules of oxygen. Okay, so binding with oxygen is a reversible process which makes it good for unloading and loading oxygen. Okay, so when the haemoglobin loads oxygen, it becomes oxyhemoglobin, and deoxyhemoglobin and oxyhemoglobin are different colours. This makes it easy to measure the percentage saturation of a sample if you ever need to know how much oxygen is in the sample. So you can do this using a colorimeter. So the more oxygen, the more saturated the haemoglobin. Okay, so here we have the general equation for the loading and unloading of oxygen. It's a reversible reaction, so I should add another arrow going backwards. And this means that the forwards direction is loading, because the end product is oxyhemoglobin. So loading oxygen means that the haemoglobin has a high affinity, and this takes place in the lungs where there is a high partial pressure of oxygen. So the same with unloading, it means that the haemoglobin has a low affinity as it doesn't bind to it anymore, so the oxygen is released, and this often takes place in the muscles where you need the oxygen for respiration of cells so they have energy. Okay, so the next part in this unit is oxygen dissociation curves. So the oxygen dissociation curve shows you the percentage saturation of haemoglobin at a partial pressure of oxygen. So for the graph, you can see that at this point there is 100% saturation, and this occurs when the oxygen has been loaded into the haemoglobin. Therefore, this occurs where there's high affinity, and this means that the oxygen is loaded in the lungs. So this point is the lungs. At this point on the curve, the percentage saturation is relatively low, and there is a low partial pressure of oxygen. This must mean that this is the muscles, due to the fact that the oxygen is being used up for cells' respiration and the haemoglobin is unloading the oxygen, so they are becoming less saturated. So you can see that the curve is not directly proportional and it's an S shape. This is because it's harder for the first oxygen molecule to bind, because the haemoglobin molecules have to change shape. However, once the first molecule has bound to the haemoglobin, the other oxygen molecules can bind more easily. So this is called cooperative bonding. Also, you can see that the top of the curve is flat for quite a while. This is because in the lungs, oxygen is constantly being brought in by ventilation, which maintains a high concentration of oxygen and so a high partial pressure of oxygen for quite a long period. Okay, so now I'll quickly go through how oxygen actually enters the tissue capillaries and how it actually binds to the haemoglobin. Okay, so first oxygen is brought in by ventilation and will be present in the alveolus. So the oxygen will bind to the haemoglobin as they enter the alveolar capillaries. It happens in the capillaries because there's a short diffusion distance, which, should know by Fick's law, increases the rate of diffusion. So the oxygen will then enter the red blood cell by diffusion, and the oxyhemoglobin retains its O2 until it reaches the capillaries. This is because arteries have too thick a wall, so they can't diffuse out. So once the oxyhemoglobin enters tissue capillaries, it diffuses out into the tissue cells for tissue cells to use for respiration. Okay, so now we'll look at how changing the conditions will change the oxygen dissociation curve. Okay, so first we'll start off with how carbon dioxide affects the dissociation curve, and this actually has a name. It's called the BIOL effect. Okay, so if this black line is our normal carbon dioxide level and this red line is a high carbon dioxide level, we can see that the curve has been shifted to the right. So the main three things you need to note is that the curve shifts to the right. This means that the hemoglobin has a lower affinity for oxygen. So more oxygen is unloaded more readily to respiring muscle cells. So the reason this happens is because active muscles are aerobically respiring more. This is because they need energy for contraction. So a waste product of anaerobic respiration is carbon dioxide, and so active muscles have a high level of carbon dioxide. Carbon dioxide, that as you should know, is acidic, so the pH lowers. And because hemoglobin is a protein, a low pH will cause it to change shape, denature and break bonds. So this means that more oxygen is unloaded. So to summarize, we can say that carbon dioxide makes hemoglobin more efficient at releasing oxygen, and that the hemoglobin must be exposed to a higher partial pressure of oxygen to become fully saturated. Okay, so now we'll quickly run over temperature. So you can see from the graph that a high temperature causes the curve to shift to the right, a low temperature causes it to shift to the left. And so, like the last slide, we can state that a high temperature does three things. Shifts the curve to the right, it means hemoglobin has a lower affinity for oxygen, and that oxygen can be unloaded more readily to respiring muscle cells. And this is the same but opposite sense for low temperature. So how can you prove this in an exam? A good thing to include in your answer is some data from the graph, and so if you are given an oxygen dissociation curve, you can draw and extrapolate the graph to compare two different curves. So for the same partial pressure of oxygen, you can see where each curve is saturated. Therefore, you can easily describe how one change in condition can affect the curve and the percentage saturation of oxygen. So for example, on this graph, at a partial pressure of 50, the hemoglobin curve with high CO2 is only 70% saturated, whereas normal CO2 is 90% saturated. This shows that at a high CO2 level, it's less saturated, so more oxygen is unloaded. So the oxygen must have a lower affinity with the hemoglobin. Okay, so different animals also have different hemoglobins. This is all to do with their survival and differences in base sequence. So for the red curve, you can see that it's shifted to the left, and this is because they live in low oxygen concentrations, so they need a higher affinity for oxygen. This means they can get saturated at a lower partial pressure. So their hemoglobin can be saturated even when there is a limited amount of oxygen available. Fetal hemoglobin also has a curve like this. Okay, so the green curve represents animals with a high respiration rate. The curve is shifted to the right, which means that they have a low affinity for oxygen, and so it unloads oxygen more readily. Oxygen is needed for respiration, so more oxygen means they can carry out respiration more readily. This is often in small animals who have high metabolic rates to maintain their temperature, so they have a faster respiration rate too. Okay, so that's the end of the video. I hope it helped. Thanks for watching and good luck!